right, so we are ready to get started here. So we had a grand total of 3 slides left in this chapter. So we're going to finish up those last 3. Um, so I had kind of given us a little bit of an introduction to ethics and what ethics are. So this definition that we get is that ethics are a set of principles of how people should be educated, treated, and respected when participating in any of the studies that we talk about. Now, I kind of mentioned that we're looking very biasedly at human based ethics. There are also ethics for working with non human animals. So that's sort of my area of expertise, which probably contributed to me completely blanking on what I wanted to say on the next slide. At least that's my excuse. So we're going to talk just very, very briefly about some of what these ethics mean or these principles of ethics mean. And then I'll talk to you a little bit more about what they mean broadly and how we actually see them in practice. So our first principle, that one of beneficence and non-malfeasance, is basically saying that you should treat people in ways that benefit them, don't cause them unnecessary harm, and ensure that your research is going to be some benefit to society. You can't do research just because you want to learn something. You have to be able to sort of put a positive spin on it or have the possibility that it will be beneficial, especially if it's something that might cause harm or discomfort for people. If your research isn't going to put anyone out, then you can get away with a little bit more of that. I'm interested in it, so I'd like to learn more. But for the most part, if there's ever any question of causing any kind of harm, you usually have to justify why this research is important and what it could benefit society once we get it done. Our second principle is the one of fidelity and responsibility. And that's basically saying that we should be trustworthy and behave responsibly, establish a relationship of trust with the people that we're going to work with, and don't break their trust by doing anything underhanded. Our principle C is integrity, which is fairly similar to principle B. So this just says that we're making an effort to be as accurate, truthful, and honest as possible. So operate with integrity. Don't lie about your data. Don't make things up. Try and keep things as precise and accurate as possible. The fourth principle is for justice. And this is where you have to treat all groups of people fairly. You have to make sure that you're selecting samples equally from the population that benefit from the potential research and be aware of biases. Now, this principle is very important for a lot of things that have been done in the past when it comes to research. So if you've ever taken a class that talked more in depth about ethics, you might have heard of the Tuskegee syphilis study. So that was a time when they were studying the effects of syphilis on a predominantly low income, mainly Black neighborhood or population group. But the research was something that would benefit all people. So that's a it's a big violation of that idea of justice. And that's one of many of the studies that you could talk about in terms of ethics that pushed us to now have this established board of rules where we know what we should follow, what we can and can't do, and where it's not just the researchers who make decisions, they have to report to those research boards to get their studies approved before they can proceed. So that's a very big one. And then the last of our principles is the respect for people's rights and dignity. This one ties massively into the idea of informed consent. So you need to be aware that people have autonomy, respect their rights, and ensure that they know enough to give informed consent. And we'll talk about informed consent again in just a moment here, but you also need to make sure that you take necessary steps to avoid coercion. So you don't want anyone to feel pressure to take part in a study. So if you ever do research with a university group, there's always that note that if you want to withdraw your consent at any time, you can walk away, no questions asked. That's no pressure added. And we'll talk about some special ethical considerations on the next slide where we need to keep those things in mind. So all of these principles, you might see them listed as the full five. More recently, I believe they've condensed into three principles that cover the exact same ideas, but in a slightly different arrangement. This is the basic idea that we're focusing on, though. So make sure that your research is going to benefit everyone, make sure that you're fair and just about it, have good intentions, do good science, don't lie about things. Um, that's basically the big message of ethics. We do have those special ethical considerations I hinted about. And this is special cases where some individuals aren't capable of giving informed consent. So, informed consent, if we kind of break down the words, it means that they give their consent, they can say that they would like to participate in the study, but they have to have all the relevant information to make an educated decision to join the study. So we could probably think of a couple of groups that might not be able to give informed consent. Children are a massive group that can't give informed consent because they're not old enough, they don't have the capacity to grasp the implications. So instead, what we'd have to do is get informed consent from their parents or guardian and just get them to agree to the study after we have approval. And this also applies for different groups. Maybe if they don't have the mental capacity to understand what's going on, then we would have to get consent from a parent or guardian. But as a researcher, you also want to make sure that you're not pressuring people to participate. Another group that actually we have to watch specifically for that intention of coercion is actually prisoners. So this is a weird one, and I spend a lot more time about this in a methods course. But for people who are in prison, there's that sort of hierarchy of power where if somebody tells them to do something, they do it, no questions asked. And so if you go in as a researcher and want to work on a population of prisoners, you have to make it clear to them that there will be no backlash, no punishment for not participating, that they're not going to be judged or anything like that, because they can feel pressure to participate simply from the environment that they're in. So we have to take extra steps if you ever want to do research on one of those sort of special groups. And there's lots of different special considerations depending on who you're studying and what you're studying, but these are sort of the biggest ones that come to mind. All right. And continuing on with our informed consent discussion. So we also have to keep in mind some of the stuff that we've talked about in designing our studies. If people know what your study is about, won't that end up biasing their behavior? Because we talked about people might act in a way that they think you want them to act, or they might act in the opposite way to be jerks. But if we want to measure their natural behavior, then telling them what the study's about before the study probably isn't the best way to do it. But that's directly in contrast to informed consent, so how do we proceed? And the way that this works is that some studies may not reveal the true nature of a study until after the participants have participated. This would be called deception. And deception of any kind needs to be part of your ethical approval. The benefits of this study need to outweigh the potential risks of not telling them absolutely everything up front. What we typically tend to see in terms of deception is more you don't tell them the specifics of the study. I've helped with some experiments. I don't really do anything with humans, but I helped someone set one up. And basically, we were going to get people to listen to black-capped chickadee songs, and they were either from Ontario or BC. But we didn't want to tell them that they were going to listen to bird songs that differed between locations. So we just said that we were going to play sounds for them on a computer and that they were going to have to put them into categories for us. So it wasn't outright lying, we just left out information that might bias their behavior. And then at the end, in order to combat any type of deception, whether it's large or small, you do what's called a debriefing. So you sit them down, you have usually a piece of paper that outlines exactly what happened in the study. So if you had a small deception, like us not telling them they were working with chickadees and that they differed on location, at the end we'd give them a piece of paper that was like, hey, those sounds that you were hearing, they were chickadee songs. They differed on population, different locations, and here's what we were looking at. Usually we'll give them a brief summary of the research. And most of the time, we'll actually give them the option to request sort of getting in touch with them when the study's done so they can figure out what happened, mostly because people tend to be curious. But in cases of larger deception, there's a whole process where you have to explain what the deception was, why they were deceived, and what information sort of needs to be corrected. So a debriefing is a really important part of any study where you don't tell them absolutely everything in advance. And so, like I said, most studies are going to go the same basic route that my chickadee example went, where you just don't tell them all the specifics, but you can still let them know the risks associated. So you're going to listen to sounds through headphones. You're going to stare at a computer screen, and there are associated potential risks with that. And that's sort of what we would outline, just so that they can give informed consent without all the details, and then we rectify it at the end. So that's the on paper what it should be, the practically of how it ends up working, and a little bit of our rationale as to why it happens. But that's a very, very brief summary of, as I said, a topic that could have an entire class of its own. So yeah, we'll leave it there because it can get a lot more complex. But we're going to move on actually to the next chapter. And so this time around, I've actually only posted the first couple of slides because I'm not positive how much we're going to get through. And I'm going to sort of decide pacing based on that. But we are quite fortunate in that we have 2 classes this week and then all next week to sort of work through this chapter, because this is one of those chapters where we're getting into a lot of information. So the first 2 chapters, chapter 1 was just sort of a general introduction to psychology. Chapter 2 is when we started learning about the specifics of science and doing research. And now we're getting into, I guess, the real content, where we're going to start talking about the biological things that underline our behavior. And so if you've taken a bio course, this will be fairly familiar. In fact, this is probably going to at least start off close to what you've seen in like high school biology. But we're going to add some complexity as we go through, so there might be some new things here, just as a disclaimer. All right. So when we're talking about the brain and the things that our brain is getting us to do, the behaviors that we're enacting, we're going to look at what is going on at the smallest level. So we're going to look at the level of individual cells, at least to start with. As we work through this chapter, we'll eventually build up into larger structures, like looking at the nervous system and the organization therein, and go from there. But we're starting with the humble neuron. So neurons are cells that convey sensory information into the brain. They carry out operations and they transmit commands to the body. Those are 3 different sort of roles, and we'll actually find that there are 3 broad categories of neurons that we can talk about. There are the sensory neurons that bring information from our body to our brains. There are the interneurons, the ones that act as go-betweens in the middle. And then there are motor neurons, the ones that go from the brain out to the body to create movement and action and things like that. So there are lots of different roles that they can do, but basically all of our movement, all of our thinking, all of the things involved in behavior are going to be driven at some level by neurons. And so we're going to talk a little bit about the base structure of neurons, maybe touch briefly on some of the variants that we have, and then how they work. So we're going to be talking about things like the cell body, the nucleus, the dendrites, axon, and axon terminals. So if you've done stuff on neurons before, those are familiar, and if not, we're going to start. All right, so this is what you would typically expect to see when we talk about neurons. Not all of them are going to look like this. In fact, most of them won't look like this, but this is a good starting point. This is a good prototype for us to figure out what neurons kind of look like. And so here we have a couple of very important structures that we're going to point out. The first being right in the middle, we have our cell body. And if we think of the fact that our neurons are cells, all cells have to have that interior where things are happening. So the cell body, which is also called the soma, is the main part of the cell. That's where all of the organelles are. That's where our mitochondria are working. That's where the nucleus is located. That's where all the stuff that makes the cell function like a normal cell is going to be located. But because these are specialized cells, they have much more than just your basic cell content. And in fact, we basically just sort of point out that they do have a nucleus and then we move on. And so then we can talk about the dendrites. So those are the little branching bits around the outside of the cell body. And the root word for dendrite actually is relating to those branches. Um, and those branches are where we are going to be receiving signals from other neurons. So the dendrites and where they're connecting into the cell body, that's where other signals can be coming in to cause sort of reactions downstream. Next, we can then look at our axon. So that's sort of that long, skinny line that's protruding off of our cell body. There's a couple of extra things here, like the myelin sheath and an axon hillock that are mentioned there, but we're going to come back to those later when we get into sort of extra functions and uses of things. For now, we just care about the axon because that is sort of the projection that allows us to transmit signals that have come in from other neurons and send them elsewhere. So you can see the little red arrows that are showing a direction of movement. So that signal is always going to transmit from the dendrites and the cell body towards the axons. So it's always that one direction. And at the end of our axons are the axon terminals. And these are, again, called a whole bunch of different things. They might be axon terminals, just terminals, axon buttons. I'm trying to think what else I've seen. There's a lot of different terms. I typically use axon terminals because it hits a bunch of those cues. But basically, they're the ends of the axon where it branches out into a couple of little, almost like fingers. And at the end, they get just a little bit wider to increase surface area so that they can send out their signal to whatever's next in that chain. So usually we look at this idea of sort of a linear relationship where we have an axon at the beginning, and then that axon communicates to the one in the middle, and then the axon in the middle, or sorry, the neuron in the middle communicates with the neuron at the end. So we always think of it as sort of a linear relationship between these neurons, but that's not always the case. It can get a lot more complex. We can have sensory information coming in from sensory receptors. So tactile receptors on our skin connect to neurons that send signals to our brain, letting us know what we feel on our skin. And then our motor neurons will actually send commands to our muscles, kind of like what's illustrated there, where it's touching a muscle fiber. So we'll usually talk about it as neuron to neuron contact, but you can have other things involved. All right. And so I already kind of jumped the gun and talked a little bit about our motor, sensory, and interneurons, but these are these definitions that go along with that description. So the motor neurons are the ones that are carrying commands from the brain to our muscles or to our organs. So that's our brain telling those muscles to move or giving our organs instructions on how to operate or things like that. Sometimes they're conscious signals, sometimes they're unconscious signals, but that will be a later problem for another chapter. Our sensory neurons are the ones that take information in from our body sensors and bring it to the brain. So they're bringing information from our outside world into the brain and the spinal cord. And when we start talking about sort of our divisions of systems, we'll talk about the central nervous system, which is the brain and spinal cord, as opposed to the peripheral nervous system, which is everything else. And then, as we said before, the interneurons are the ones that go between the motor neurons and the sensory neurons. So they connect one neuron to another in some part of the brain or spinal column. So our brains are composed of lots and lots of interneurons that are acting as sort of go-betweens and then taking the information that our sensory neurons bring in and sending out our decision to the motor neurons. So they all work together. And so, as you can imagine, our behavior is fairly complex, so there's usually a lot more steps involved, but we're keeping it as simplistic as possible. And this slide here is more of a for your information as opposed to getting anything huge off of it. I just wanted to show you that when I talked about that neuron we were showing being a prototype that gives us a good idea of what neurons are, but not all neurons, these are some of the other types of neurons that we can have. So the top of the screen, we have a unipolar neuron, and that's a sensory neuron where our dendrites actually connect directly to our axon and the cell body kind of goes off on the side. That's a weird one, but it's specialized for really rapid communication. So in that case, because that signal doesn't have to go through the cell body, it goes a little bit faster. So that's why that's usually a sensory neuron for that kind of an orientation. We can also have another kind of sensory neuron here, a bipolar sensory neuron. And that one has one set of dendrites branching off, and then one set of the one axon, as always, with the cell body in the middle. And that's going to be sort of semi-quick, but not super quick, going from one place to one place, more or less. And then the interneurons tend to be a little bit more chaotic. They tend to be a lot less, here are the dendrites and here's the axon. It tends to be more spread out like that, which is fun. And then what we were looking at as our stereotypical neuron is actually called a multipolar motor neuron. So it would receive information from a bunch of places because it has lots of those dendrites at the top, but still just the one axon leading to its place. So just a little bit of an example of some of the variety that's there. We're going to basically continue assuming that all of them look more or less like our multipolar motor neuron, but I just want to make you aware of the possibilities. Okay, so now we're going to step back into our biology shoes, and hopefully you remember a little bit about the cell membrane. We're going to keep it, again, not as intense as if you've done cell bio, but you might recall that our cell membranes are made up of a phospholipid bilayer. Basically, there's a phosphate head with the two lipid tails. So that's what each of those little red things with the two legs on it are. So that would be a phospholipid, and they're going to orient together in a particular way based on the properties of the molecule. And so the way that this works is the head, that red ball, is going to be hydrophilic. And from the root of that, hydro, we're talking about water, and philic means that it is water loving. So that phosphate head will dissolve in water. And so when we have our intercellular and extracellular fluid, all of the liquid that's around and in our cells, the phosphate head is okay with that. It doesn't mind being near the water. So these phospholipids are going to orient so that the heads are touching water. In contrast, those lipid tails are hydrophobic, meaning they're afraid of water. So they want to get as far away from water as possible. So they're going to orient so that they're facing away from all of the liquid that's outside the cell or inside the cell. And that's how we end up with this double layer of these phospholipids where all of the tails are on the inside and they're keeping the water out, and all of the heads are on the outside and they're fine with touching the water. Now, this difference in sort of solubility is actually due to a difference in like charges and some of the properties of those cells. But all we care about is the fact that having that kind of an arrangement means that it's very, very difficult for any molecule to pass across this layer because the molecule would have to be either water soluble to pass through the phosphate heads, or it would have to be water insoluble to pass through the tails, but it kind of has to be both, and you can't be both. You have to be one or the other. So most things cannot pass through this layer because of those properties. And that's what lets us actually have all the stuff we're going to talk about today. So this barrier keeps what's outside of the cell outside and what's inside the cell inside until we choose to release it. And so, how might we go about doing that? And there's a couple of ways that you can move molecules across this membrane. So the first that we're going to talk about are protein channels. And again, stuff has a bunch of different names. You might call it a protein ion channel or just an ion channel. What these do, these are proteins. They're made out of protein, and they fit into that phospholipid bilayer. They have parts that can associate with the lipids. They have parts that don't mind the water, and they can slot in, and they act as a bridge across that membrane. So we can allow things that are outside the cell to come into the cell, or things that are in the cell to go out of the cell. And we have different kinds of channels that work with different kinds of ions or different molecules. So they'll only allow certain types of molecules to travel in a particular direction. So we're going to be talking about sodium ion channels, and they're only going to allow sodium out of the cell. Or we'll talk about potassium ion channels, and they let potassium ions into the cell. And that's really important. So these can be turned on or off. When they're turned, or this process of sort of turning them on and off is called being gated. So they can be electrically gated or chemically gated. And that basically means that there's a switch that can turn them on or off. And that switch is either a chemical or an electrical charge, which is really important, again, for later. But that's our basic setup there. Next, we're going to hit a couple of terms, and these are all fairly closely related to one another. So we're going to go through them nice and slowly. So having this membrane in between and controlling what can move inside and outside, meaning that there isn't free movement across the membrane, it means that we can have a state of polarization. So this is a state where there's a difference in electrical charge between the inside and the outside of the neuron. So we say that the cell membrane is polarized. There's polarization across the membrane if there's a difference in ion charges inside and outside. And so that'll be important in a minute, but having that difference in charges, having that polarization across the membrane actually means that we've stored energy across the membrane. Because you might remember from sort of bio and chem that molecules would like to move around so that they're away from other molecules that are the same kind. Positives don't like to be next to positives, negatives don't like to be next to negatives, they'd like to spread out. So by forcing them to stay in a place they don't necessarily want to be, we're storing energy there. So we can say that there's an electrical potential or an ionic potential across our membrane. So that potential will be important. Actually, we can say that part now. We can go to our last point, which is saying that we have a resting potential, which is where the cell tends to sit, where it's at rest, where it would like to exist, where there's a difference in charge between the inside and outside of that membrane when the neuron is sort of just sitting. So resting potential, we're saying that there's potential energy stored across that membrane when the cell's at rest. And we're going to talk about just in a second about how it sets up that potential, how it sort of moves stuff around so that it has this energy stored, kind of like a battery, to be able to use when it needs it. And if we're talking about the intensity of that charge across the membrane, we might talk about it in terms of voltage. The voltage lets us measure the difference in electrical charge between 2 points. So when we report the potential energy, that electric potential across the membrane, we're going to report it in terms of actually millivolts. So we don't have enough energy to measure it in volts proper. So this is kind of like millimeters versus meters. So we have small volts, And so that just means that there's a slight charge across the membrane, and a voltage can only be measured as a difference between 2 points. We don't get an absolute voltage. What we care about is getting that difference between the inside and the outside. Okay, so the next of our things we want to mention are our ions. We've already, again, kind of mentioned them a little bit. So ions are atoms that have lost or gained electrons. So they'll have a symbol, a plus or a minus, that indicates that they've lost electrons, and electrons are negatively charged. So when you lose electrons, you are net positive. But if you've gained electrons, if you've picked up extra negatives, then you are more negative. So by indicating if something is positively or negatively charged, it's just telling us what's happened with the electrons floating around. So for our positive ions, we're only going to talk about some of them. We're mostly going to focus in on sodium. So Na plus is a sodium ion that has lost one electron. We're also going to talk about potassium, which is K, and it has also lost one electron, so it's plus. We might see in a couple of slides, I think I bring in calcium, which is Ca2 plus, and that just means that it's lost 2 electrons, but that'll be relevant later on. For our negatives, we have chlorine with an Cl minus, so it gained one extra electron. And then we have a weird one, and this is called A minus. And if you're familiar with the periodic table, we don't have an element that is just A. What that is sort of a shorthand for a group of organic anions, basically generic molecules that have an extra negative charge to them. So we kind of hand wave that. So if you see A minuses in our pictures, that's just a blanket covering of stuff that is not super relevant for us. And so if we wanted to look at what our cell surface might look like, we have a couple of, or we have a channel in the middle that might allow ions to pass through, and we have differing concentrations of those ions inside versus outside. So we have more of the red ones on the outside and more of the green ones on the inside, and theoretically that's setting up that resting potential. And so just for a little bit of an illustration of what's making these ions move around, this again might be a little bit more info than you need. And I typically try and avoid PowerPoint animations, but this is one of those times where I just took a deep breath and included them anyways. But so I kind of already mentioned that these ions want to move around based on spreading out, getting away from things and getting away from themselves. So we can talk about two forces. The first is a force of diffusion. So the tendency for ions to move through the membrane to a side that has less concentration. So potassium ions don't want to all be on one side. They'd really like to get to the other side. They want to get away from each other because they'd like to spread out. They'd like to diffuse into as much space as possible. So that gray line in the middle would be like the membrane. For this example, we're showing what they would do if they could move, not what they're forced to do when our membrane blocks them. But there's tension between them and they like to spread out. So they're going to move so that they are more distant from one another. They diffuse and spread out. So that one is driven by sort of, they don't want to be near similar ions. They want to get away from the same things. And that's closely related to another principle, which is electrostatic pressure. And this is basically the same concept, but driven more by the electrical charge. So it's not that potassium ions don't want to be around potassiums, they don't want to be around any positively charged ions. And our chlorines, they're negative, they don't want to be around other negative ions. But along with that, not just wanting to get away from like charges, they're also attracted to opposite charges. So the chlorines would like to be on the positive side because they'd like to equal out. So they'd like to bring that charge back to zero. They'd like to get to a place where they're basically happy. And so for these, they're going to sort of swap across and get to a general negative charge. This is what they're aiming for. And these two forces are what are actually setting up that electric potential across our membrane. because they want to be on the opposite side, but when our membrane stands between them, it's storing that energy. It's making them stay somewhere where they're not content. So when we release that energy, when we open up and allow them to move the way that we want, we can actually harvest or harness is the word I wanted. We can harness that energy, that movement to accomplish things. So that's the setup that we're putting together. That clock is broken, so that's not helpful. All right, I think we have just a little bit more time here. So I've talked about the fact that our membrane is holding this charge in place. There's a voltage across our membrane, our resting potential is established, and things are in sort of in flux. They would like to move but can't. How does this kind of a setup exist? If they want to be at no charge, if they want to move around and be even inside and out, what is setting up this stored energy? And this is where we bring in our sodium potassium pump. So when I was talking about this image at first, the thing in the middle is probably one of our ion channels. Those are the ones that can open up and let ions in or out of the cell. What we care about now are actually the two things on the outside that look kind of like hot dog buns with little half circles cut out of them. Those are the sodium potassium pumps. And so from the name, we can assume they work with both sodium, which was our Na plus, and our potassium, which is K plus, and they're going to pump or move them around. So pumps are proteins, again, large protein molecules. They are going to be embedded across our cell membrane again to facilitate movement, but they're actually powered. So unlike our channels that once they're turned on, the channels just allow things to flow in or out as they want to. The pumps are actively using energy, they're burning ATP in order to push these ions into that sort of disordered state. So they're creating that charge by moving sodium ions through the cell membrane to the outside. So, oops, wrong way. So on this side, we have our red Na pluses. They are pumped outward. And with every sort of pump of this pump, every beat that it does, it will dump 3 of those sodium ions outside. And when it sort of retracts, when it does the other way, so if you think of a pump, it's going to go one way and then the other. So it's going to push outside, drop the 3 sodium ions. When it's pushing inside, it's actually pulling in 3 of our potassium ions, or sorry, 2 of our potassium ions. Make sure I get all the moving parts right. So what we're having is a net movement of, with each pump, 1 extra positive ion is going out, because we're moving 3 positives out and only bringing 2 back in. So we're setting up an overall negative charge where the inside is going to be comparatively negative if we're comparing it to the outside, because we're having less of those positive charges inside. So again, we're not referring to the voltage as an absolute because we're only moving positive ions. But what we're getting is that there are more positives outside and less inside. So the inside is effectively negatively charged. And so then we can talk about how once we've established that tension across the membrane, what happens when we then trigger those ion channels? What happens when we trigger them to open? And I already dropped this information saying that they can be chemically or electrically gated, which just means that they can be switched on using either chemicals or an electrical charge. And then they can let those things flow back where they want to go. So we're actually going to leave it at this point because I think we've set it up pretty good as to how we've gotten to our resting potential. And then in the next class, we'll talk about what happens when we release it, when we allow them to flow back to where they want to be. So yeah, we'll review a little bit next time and then start with what happens next.